kind of a proof of concept of how to improve the docking for antibody-antigen interaction. So this is within the context of my thesis, where I'm co-supervised by Fernando Barroso, that's here from USP, from Universidade de São Paulo, and by Catherine Etchebest at Université Paris-Cité. So antibodies are very useful therapeutical tools and also diagnostic tools nowadays. And they can be engineered, as we saw during the week, yesterday, for example, so by different means. And usually, when you try to engineer or design an antibody, you want to have some information, some level of information where the antibody interacts with your antigen. So the antigen is going to be the target, whatever the target, okay? So you want a level of information about the surface. And this we can call the interface or the paratope-epitope association. So the paratope is going to be the antibody part that is in contact with the antigen, and the antigen part is called the epitope. Just a bit of vocabulary. So you can predict, actually, you can study this interaction process by predicting it, for example, with docking tools. So this is basically methods that try to sample the conformational space between the two molecules that you give, and they will give you conformations. This conformation we can call docking models also. Okay? So it works like this. You give the separated molecules, so you have your two proteins, and you give that to the docking tool, and it's going to generate a number of conformations. In the blind, usually, you generate a lot of poses, a lot of docking models, like 10,000, something like this. Here it's 3,000. And from this pool of conformation, you're going to have a great diversity of conformation. So lots of conformation and lots of variability. So this is the first step, but there is a very important second step. You have this ensemble of poses. Well, you have to select certain candidates. You cannot go forward for your engineering process with 10,000 poses, right? So usually these docking tools, they have a way of ranking by their scoring function that's usually physics-based. And they will provide a rank. So these are the most likely states for your conformation, and these are the less likely. And we can actually, we can evaluate these docking tools, and we can evaluate them by taking actual crystal structure of complexes that are already solved, and we give to the docking tool the separated molecules, and we see if within the models that it provides, we can find some models that are very, very close or near native-like. So these are models that are very close to the crystal structure. So we measure that by some criterions. So for example, the native fraction of contacts between the native state and the model that is proposed, or the interatomic distance when you overlap, you align the two complexes. So this is the IRMSD. So we use this to qualify if a model is a good quality model, which we would say it's a near native model or not in a benchmark situation. Just a quick example, visual. Here you have a reference. Here it's a crystal structure of an antibody in the two colors here and the antigen in green. And here you have a native model. So you can see that between the two, like visually, they are very similar. And also this is highlighted by the metrics. So we have IRMSD that is really low. You want that to be as close to 0. And the contacts are 100% conserved. So you can see that it's a this is a near native model, and this is what we want that the docking tool provides. Okay? And here it's an example of a non-native model. So you can see visually that the conformation is off, and this is also highlighted by the criterions that we selected, where we have a higher MSD and 0 native contacts. that remains. Okay, so that's what you don't want at the top selection you don't want to select those models. So I've talked about near-native models, and you can ask, oh, but you have already the crystal structure, so why bother predicting it if you just use the crystal structure? So let me answer by explaining why it's important to predict near-native states in a benchmark situation. So let's see two situations. When, for example, you have the reference complex, where is it? When you have the reference complex and you test a docking tool, it's not able to propose near-native states to you. So it doesn't provide useful confirmation. Then, when you don't have the reference complex, in most situations, actually, you are in the blind. You're trying to predict something, so you don't have the answer. Well, then you will have less confidence that the docked states that are provided by the tool are likely. In the other hand, if the tool is able to propose near-native models to you, in a real situation, you will have a better confidence that these docked states are likely. Okay? So, how does, so let's benchmark then. Let's see how does docking performs an antibody-antigen interaction specifically. So here it's a figure from a paper from Bonvin in 2020. It's the developer of AbAg, which is a docking tool. And they compared different docking tools for a benchmark set, a subset of structures, of complex structures of antibody-antigen interaction. And the key message here is that these tools are able to provide useful confirmations. However, during the ranking, they don't rank them in the top position, which can be a problem. So they can provide things that are useful, but the scoring needs a bit more work. And why is that? Because these tools usually have, historically have been developed for general protein-protein interaction, so non-immune and immune protein-protein interaction. And the two types of interaction, immune and non-immune, differ from certain properties. So for example, they have a smaller interaction surface, less shape complementarity. Some amino acids are present or absent in the two interfaces. So that might lead to these tools performing less well. So in brief, we've seen that predicting accurately the interface between an antigen and an antibody is useful for engineering, for example. Docking is a method to provide confirmations, to study this interaction and to provide predictions, and that we can benchmark those methods, and that we saw that the ranking of these models can be improved. Okay, so our objective is not to make a new docking tool, is to use the best docking tools that are available. Here we're gonna use AbAg. And it's instead to create a scoring function that's gonna be specific for antibody-antigen interaction in order to re-rank the docking models that are provided by the tool and to make the near-native models appear in the top, more in the top selection, okay? So we need to start by building a data set. So here I'm going to be very quick, but this is a very important step. So it's 845 reference complexes, and we have a process of filtering these results. Yeah, we have a process of filtering, but we're going to jump over for time purposes, but this is a very important step. And from this dataset, we've made a bunch of basic observations. So the first one, it was to analyze the amino acid propensities, the amino acid frequencies at the interface. And we analyzed this for antibodies, that's Fab, antigens, and of our dataset for sure, and non-immune protein-protein interactions from another dataset. And what we can see is that there are some specificities, especially in the antibody side. So there are much more tyrosines and serine in the interface. So that's an interesting observation. A second thing that we can observe, and that's underlined by the following question, is so the antibody is binding in this region, which is the epitope, and you can ask, oh, is this region specific? Has this region some features that are different from the rest of the antigen surface and makes the antibody binds to it? So we can, again, look at amino acid propensities very simply. And we can see that some amino acids that are charged also are overrepresented at the epitope surface. So this suggests two things, that amino acid propensity can be used probably to draw our scoring function, to draw a distinction between a good paratope epitope interface and another. And also that the electrostatic contribution might be important also because we see an overrepresentation of amino acids that have a charge. Okay, so we've built a data set, we've made some observations, and we can use these my God. We can use this observation probably to draw our scoring function, to guide our scoring function. Okay, so how does the scoring function works? I'm going to go very briefly, but because I'm running out of time, but you start with the atomic representation of your docking model that you want to score, sorry. And you have to choose a representation for your scoring function. Then you score your docking model. So the representation is based on our observations. So we're going to use amino acid frequencies at the interface as a feature vector and also accessibility to the solvent. Then we choose a way to predict. So a classification task here, it's going to be drawing between native and non-native, predicting between native and non-native. And finally, we choose a way of a scoring function, the actual function. So here is going to be a neural network that we're going to train and then test. So we've seen this week how it works. So I'm going to go over this. But for testing purposes, you give it unseen data, so this is very important, and you can evaluate it by some metrics, like for example accuracy, F1 score. But for this type of problems, MCC, Matthew cross correlation coefficient, is very good. It's maybe the better one. And you check if your trained neural network was able to generalize on your test on your trained set. Okay, so this is the architecture, the pipeline of our scoring function. And it's not super interesting, it's very basic. We take our docking model, we get our data representation, and we give it to 2 fully collected layers and 1 output layer. So it's a really sigmoid output layer, and it's really simple. It outputs a score between 0 and 1. For the epitope and the paratope separately, we average the score and we get our final score. Okay, so let's see the results. So these figures shows that if you were to rank your pool of models by different scoring functions, what would be the rate of near-native model in the top X models? Okay, so what you want is the highest thing, the highest score, the highest rate, close to 1. In the top 1, you always want to select near-native states, and then it goes down. Okay, so you can see that we have the best case scenario. This is a hypothetical scoring function in blue. You cannot beat that. It's artificial. Then we have the apism. See, this is the amino acid propensity interface scoring model that we built. And we have the Haddock score, if you were to rank with the Haddock score. And then finally, you have the average near-native rate. So if you were ranking by random sampling. So you can see that Haddock is much better than random sampling, and that we improved on the scoring function of Haddock. So we have also built other models that are better than this one, but to summarize, so we've made observations on the interface properties of antibody-antigen interaction, and this information was used to discriminate likely states from non-likely states. So this is a very simple step to build your scoring function to your specific problem, to improve, for example, here, antibody engineering, the antibody engineering process. Thank you for listening, and sorry for the extra time.
Questions?
When you show the graph of the pose, the scoring function.
Yeah.
Well, the best case scenario is that the top one structure is the best ranked.
This is it's the best case scenario.
Yeah. So you have some actually a few ones to have the best. So you have a little slope. Why is that? You must I don't know five structures to have the best one.
Why it increase? Okay.
Yeah.
Because it's more difficult to get it right for one structure than to get it right for a bigger number. That's why.
Okay, other questions? If not, we can thank our speaker again and move on to the next one. Yeah. Thank you. Okay, please.
Now? Okay. So this is I think it's my first English presentation, so good luck for you to understand me. And I drink a lot of caffeine, so I'm a little fired, but we can fire together. I prepared this joke. So I represent Kaistella, jeonii, esterase, conformational dynamics, but binding with PET molecule study. I am Arthur, and my advisor was Professor Dr. Márcio Dorn, and co-advisor Dr. Ederson Salles. I think you might know him. So plastic pollution is a big issue globally because plastic residues are found almost in every environment and can be found also in some human tissues. And by 2050, by 12 billion tons of plastic will be found in environment and landfills. So this is kind of a big problem. And PET plastic is the most used kind of plastic used today. And the most used form of mitigate this problem is almost recycling, which is not a very efficient way of mitigating this. So bioremediation is another way to mitigate this issue and get rid of plastic pollution. So in 2016, it was described enzyme capable of degrading PET and called ICTP-SAIFR, and today is the most studied enzyme for this PET degradation. It is the it is the red one here. And in 2022, a new enzyme, esterase enzyme capable of degrading PET, was described and named PET30, and its esterase was able to degrade PET in a cold environment. That is, this enzyme has a low sequence identity with ICTP-SAIFR, but shares a very similar 3D structure with this enzyme. So we can see in red, in green, the PET30, and in red, the ICTP-SAIFR. We can see that it is a very similar 3D structure. Also, both of these enzymes has the same catalytic triad here, composed of aspartic acid, histidine, and serine. And the approximation of these three amino acids produce nucleophilic attack on PET, enabling the breaking of this molecule. Also, PET30 does not have a disulfide bond on the catalytic pocket here that ICTP-SAIFR has. And this disulfide bond is in two coils that the serine and histidine of the catalytic triad are present. So it kind of attached these two structures and maintaining these amino acids closed. Okay. So the aim of this study was to apply molecular dynamics to understand the PET30 and PET molecule interaction and compare with ICTP-SAIFR literature. To do this, we model a trimer molecule of PET on Marvin JS, and we get the parameters using LigParGAN. The protein structure was obtained in PDB under the section 7PZJ, and we modeled the structure to fill some amino acid that was missing. We validate the structure in model probability, and after validation, we did an ensemble docking procedure in Vina, using Vina. So the first step was docking in Vina, and after that, we did a molecular dynamic simulation of 100 nanoseconds, and we get the most divergent conformations based on the root mean square deviation of the catalytic pocket. And then we did another docking procedure and selecting the best interaction with the structure. So after obtaining the final docking structure, we did a molecular dynamic simulation dividing in two systems. One is a PET30, only PET30, free, without PET molecule. And the another was a PET30 bound to PET molecule. Each system was composed of 5 replicas and with 1 microsecond of simulation each, giving a total of 10 microseconds. The molecular docking result I show here. This is the final docking structure. The PET molecule is right here. And after just a few nanoseconds of simulations, the PET molecule assumes this pose. And this pose correlates with the PET interaction with ICTP-SAIFR literature. So it was a good pose for us. This is a video of our replicas with the PET30 bound with PET molecule. The first one here, we see that the PET PET molecule stays in the pocket but goes away and at some point and then returns. These two, the replicas 2 and 5, the PET molecule goes away of the catalytic pocket and do not return. Then replicas 3 and 4, the PET molecule stays bound in the catalytic pocket during all the simulation. As we know the catalytic triad conformation, we measure the distance between the residues. This is the distances of the PET30 free, and we see that the PET, these residues, histidine and serine, the histidine and aspartic acid, kind of do not stay together or approximate, which is different from the PET bound that in replicas, especially in replica 3, these two amino acids get close together. This is also observable in the serine and histidine, that is another amino acid, when PET33 does not get together. But in the bound system, they approximate in the replica 3. We also measure the distance of the amino acids that are in the same position of the disulfide bond of PET-ASI. And we noticed that when the PET molecule is bound in the catalytic pocket, they do not stay in the distance of disulfide bond. So it seems like the PET molecules induces force in this catalytic pocket and provokes that these two amino acids get away from each other. Providing a which indicates that the two amino acids that which indicates impact on the formation of a catalytic triad conformation. Okay. This is the PCA plot where here is the PET33 system. We see that so let me start again. This PCR was based on the Cartesian coordinates of the backbone of the protein, and in the free PET30, the system assumes like different conformations, navigating around more divergent structures. But when the PET30 is bound, here the protein assumes a more specific conformations, leading to a more favorable interactions with PET30. This is it. So in this work, our molecular dynamics simulation reveals a favorable interaction with PET molecule, but we show difference with PET-ASI, and we give insights for the impact of the lack of disulfide bond on PET30. Also, we get insights with the interaction mechanisms of PET30 and PET molecule, and shed light for PET30 adaptation to cold environments. So this was part of my bachelor final thesis. It's a published paper. You can access under this QR code. And thank you.
Thank you very much. Other questions?
Just can you go back on the slide with the PCAs? No,
okay.
The last one. So the left one is without the binder. So it's not binded.
Yeah.
And this one is binded.
Yeah.
All the time.
No, it's all the replicas together. Okay.
all the replicates where it's bound.
No, even the one that were not bound, like here, we
You
put together.
just overlapped everything.
Yeah.
Okay. Oh, so that was my
Okay.
question. Okay, cool. And so can you just go back on the PCAs?
Oh.
And so the left part here on like the cluster that is about -4, -1. Yeah.
Here?
This corresponds to which type of conformation for your, because it's very separated from the rest.
Yeah. Probably we will need probably more time of simulation or more replicas to get these clusters together. Okay. But the conformation that are more similar to a catalytic conformation is this
the
one.
right one.
Yeah. So this one is probably one of the one that the PET goes away of the catalytic pocket.
Okay. Thank you.
Okay. Yeah.
More questions?
So thanks, Artur. I was wondering, how did you define the length of your simulation? Because you said that the replicas could be quite short. I mean, how did you define?
No, it was a good question. Let me think.
Take our time.
It was like based on the literature of the simulation of the ICTP-SAIFR. So we get the average of the simulation of ICTP-SAIFR, and we try to execute this time on our system. So it was that.
But wouldn't it be useful to take a look on
What?
Wouldn't it be useful if you take a look on the data from the simulation, I mean, to define how long you're going to run?
From what?
I mean, to look at the data, like RMSDs, the energy, this kind of things.
Yeah, we look. So I should have put the graphs, but the system has the RMSD converged. So very, very soon, actually, I think 100 nanoseconds in our RMSD. And I don't know how to.
Okay, we can discuss later. No
Yeah,
problem. Yeah.
Sorry.
No, it's fine. Thank you.
You're welcome. Yeah.
Are there more questions? Do your results allow you to say which one of the two variants works better?
What?
Which one of the two works better? Do your results tell you anything about that?
Actually, I do not work with variants, so it's just a free
No,
system.
I mean PIT30 or ICTP-SAIFR.
Oh, like ICTP-SAIFR has a more efficient catalysis because it's more adapted to PET.
Right.
But PET30 is not the main. It's not really adapted to PET. It's like a evolutionary step for efficient catalysis of PET molecule. So
okay.
we have ICTP-SAIFR that is adapted specific for PET, but PET30 seems that it's evolving to do this. So it's not specific for PET molecule.
I see. Okay.
But PET30 can break the PET molecule in lower temperatures, which ICTP-SAIFR cannot.
Right.
Yeah. So this is why we simulate the PET30, because we want to understand what enables this enzyme to do this.
Right.
Yeah.
Okay. Thanks.
Thanks.
No more questions? Then we thank Arthur again and move on to our next speaker.
can you, yeah. Yeah.
Okay, our next speaker is Fausta. Please go ahead.
Okay. So good afternoon, everyone. And I'm Fausta Desantis. And for those of you that didn't have the chance to know yet, I'm a PhD student at Italian Institute of Technology based in Rome. And today I will show you some parts of my PhD project. I basically deal with in silico assessment of protein-protein interactions. And today I will show you specifically some results related to a disease that is called antibody-antigen amyloidosis. So before I go deeper into the details of this part of the job, I will provide a small overview of the disease we're talking about. So antibody-antigen amyloidosis is the most common type spread of systemic amyloidosis, which means that several tissues could be involved that strongly affecting the quality and expectancy of life of the affected patients, especially because of the deposition of fibrils in important organs such as the heart and the kidney, which will go to failure. So it is due to the aggregation of overproduced light chains by dysfunctional B cells. And as in any aggregation pathway in an aggregating disease, we have a sigmoidal trend in terms of the aggregating species, characterized by a lag phase, where first you have just dimers, the formation of oligomeric species in the nucleation phase, and finally an elongation phase where you have the fibrils formed and deposited. So the shift from one phase to the other is driven by conformational changes, which are so transient that it's very difficult to be explored experimentally. And so this is why computation could come at help, and so why I have a role here. So what mainly drives conformational changes is mutations. But actually, in principle, mutations do not necessarily need to be the villain in this story, because you both might have germline and mutant sequences that are related to monomeric fold, which can actually be in thermal equilibrium with native dimers. We call them native because they are not involved in any pathological scenario. So you basically might have monomers that dimerize, and then the dimerization is disrupted, and this doesn't cause any kind of disease. But we also know that sometimes mutations like to mess up things around. And so it is rare, but still not impossible, that mutations will cause some misfolding events into the chain. And such misfolding will make the chain more prone to seize other misfolded chains, thus causing a non-native dimer, a pathogenic dimer, thus beginning all the chain of events that I just spoke you about. So, but what makes this disease particularly special is the fact that even though immunoglobulins have a very conserved structure, there are some regions that on their surface that are highly prone to mutation, which is also what makes our immune system so efficient and versatile. But this also means that each patient might develop their own mutations, thus making this disease strongly patient-specific and hardly to be tackled on a wider scale. So what I did was to focus on the lag phase of a patient-derived light chain, so I considered a case study in the literature, and focus on its lag phase, as I just told you, and try to understand whether I could obtain the structure of a putative non-native dimer. So the case study I considered was very well experimentally analyzed in terms of aggregation dynamics and structures. So here you have the cartoon representation of the crystal structure of the mutant on the right, depicted in red, which was extracted from the belly tissue of a patient. The experimentalists were then able to retrieve the sequence of a germline and express it so that they could obtain the crystal structure as well. So these are the main characters of this story for now. These two guys differ from 11 mutations, most of which appear in the framework, which is already a hint of the abnormality of these light chains, because the framework is not supposed to be changed. You know, it's supposed to be quite conserved. So what I did was to perform 10 microseconds long all-atom molecular dynamic simulation for 3 replicas of each monomer, so that I ended up with 30 microseconds simulation, my simulation. So here you have very classic parameters for post-MD analysis, the room square deviation. There is this is the density distribution of the room square deviation. And as you can see, the mutant undergoes larger conformational changes than the germline and explores a part of the conformational change, or the conformational space that is not achieved by the germline. So this clearly hints to some kind of misfolding in the light chain, in the mutant and pathogenic light chain. So to go deeper and to understand what was actually happening and to characterize this misfolding, we assessed the solvation properties of the proteins involved. To do so, we performed an indirect assessment using a hydropathy scale that was developed by my team, which assigns a score, an index to each residue. And this is molecular dynamics based. And according to our scale, so the lower the index, the most hydrophobic the residue. So according to our scale, azuleucine is the most hydrophobic residue, whereas asparagine is the most hydrophilic one. So we performed an analysis of the accessible to the solvent area against the mean hydropathy per residue, averaging the results per each residue in the sequence on the dynamics. And looking at the top left corner of these scatter plots, which actually refer to the most exposed residues and the least hydrophilic ones, you can see that the mutant displays some residues that remain instead buried in the structure of the germline. And these residues, which are anacylus in position 52 and a phenylalanine in position 66, happen to be hydrophobic residues. So here you have a representation of how the structure changes and how these residues are actually visibly exposed by the end of the dynamics of the mutant. And so thinking of the very nature of these residues, these are hydrophobic, so we speculated that actually they will not be so happy to be surrounded by a solvent, and so that they may try to seek shelter from the solvent by forming an interface, a new interface, which is actually non-native, like non-physiological. And so we said, okay, given this information, we can actually try to look for a dimer based on this consideration. To do so, we first perform a hierarchical clustering based on root mean square deviation as metrics of the part of the dynamics that in the mutant that show the major conformational changes. Here we have the abundances of structures in each cluster. We consider the centroids. And so this is not a pentagram for any dark magic. This is just a schematic of the docking combination that we try to make. We used the AbAg web server. And so we consider all the possible combination of centroid structures and required AbAg to build the interface in such a way that one of the residues that were exposed were considered. So in this, so we have the case in which an acyleucine 52 on one side and an acyleucine 52 on the other side, the case in which we have a phenylalanine 66 on one side and a phenylalanine 66 on the other side, and the mixed cases. So considering all the possible combination of central structures to be docked and interface composition, we ended up with 55 docked structures. And we performed 50 nanoseconds long molecular dynamic simulation on each of them. So here you have an analysis of these dynamics. We focused on the fraction of conserved contacts and the interface and the interface root mean square deviation. And actually, it's clear that the dimers that whose interfaces are mostly composed of acyleucine 52 on both sides, display the highest fractional conserved contacts and the least root mean square deviation. So apparently, the most stable pathogenic complex must reside in this group of proteins, group of dimers. So we restricted our analysis to this group and looked at the interface Lennard-Jones interactions, which we previously, in another work, demonstrated to be dominating in stabilizing binding. And as you can see, there is a specific complex that displayed a high favorable contribution to this energy. And so we extended the dynamics of these dimers to 1 microsecond. And actually, Lennard-Jones interactions is very stable, so we can say that this is quite a stable dimer, and it's actually a good candidate to be considered a pathogenic dimer at the beginning of the aggregation pathway. So what I plan to do, what I'm doing now. So first, I would like to, in the future, I would like to observe, to shift my focus on oligomerization. And to do so, I would like to try to use coarse graining, such as Calvados and Martini, that I've been trained to use in the lab of Professor Christian Lindorf Larsen in Copenhagen. who I thank even if he's not here. And moreover, what I'm doing now, actually we already have some results that I couldn't show you today. I'm trying to design a peptide to pair dimerization in principle. So the protocol, the pipeline that we're using is this. So we scan the interface, try to understand which are the most relevant residues at the interface for binding based on the number of contacts. So once we have this sequence that could be a putative possible peptide, we perform an optimization. We optimize the peptide through mutagenesis Monte Carlo protocol. And once we have the peptide, the idea is to try to perform docking to see whether it's actually it's competitive against the dimerization with two proteins, two light chains. For now, what I can tell you is that actually this peptide has higher affinity to the interface than the whole interface. Like the peptide is more affinity with a chain rather than the same chain to the other chain. So it's a good result, hopefully. Of course, a lot of refinement is needed, and the path is long, but still. So before I leave you, let me thank my team, which is not here, but is always so supported, and of course, all of you for your attention, and Fernando for giving me the chance to be here to share with you my job. Thank you.
Thank you very much. Questions?
Thanks, Fausta. Very nice talk. I was just wondering, why just the light chain, not the heavy chain as well?
Yes, because the light chains are being overproduced, not the light chains. Specifically, the fibrils have been found to be mostly composed of the variable domain of the light chain, so it's even more restricted, the thing. But actually, also the constant domain you could be found, it's very heterogeneous, so it's very difficult to characterize in a very, unilateral way, the problem. Also, there is also very little literature about it. So, it's quite a rare disease, not very tackled. So.
But have you ever tried to include the heavy chain and run the calculations to see if
the heavy chain is never involved. It's the light chains that are being overproduced and then aggregate. But in general, it's more complicated than it could be. Than it may seem. Also, in terms of the light chain, there are different contributions to the fibril formation. That was the point.
I'm curious because, for instance, when we're dealing with antibodies, antibodies that we try to optimize, it seems that we can have very nice results only using the heavy chain. Okay. So it seems that the light chain is there just to stabilize the whole complex.
Yes, but the heavy chains are not involved in the pathogenic process because they are not the parts of the antibody that are being overproduced. Mm-hmm. So you have a concentration of light chains that at some point will also rise up in the disease. It's not the heavy chains, just the light chains. And sometimes just the variable domain, sometimes also the constant domain, and yeah.
Okay, see. Well, thank you.
Thank you.
Are there other questions? No? Yeah.
Just can you go over the docking process?
Yeah.
You tried different combination of docking.
Yeah.
And you used HADOC, as
Yeah.
I said. And so here we are talking more protein-protein interaction, more hydrophobic type of interaction, if I understood correctly.
No,
it's the frameworks that are interacting.
In which the framework of the light chain?
On both light chains, it's not the CDRs, for example.
Actually, I didn't check what actually the position of these residues within the framework or the CDR. But basically, we just required HADOC. Okay, these are the residues that we want to be driving the interaction. So please use them.
Okay, and so you constrained during the docking. You constrained these positions.
In HADOC you can just state which residues are you really want to be there for. So this is what I did.
Okay, and during the selections of models, because you generated a number of
Yeah,
models,
okay.
you verified.
So AbAg provides like a scoring, like not just one structure. So we used since AbAg also provides a parameter which is a calculated binding affinity,
Yes.
we used that parameter to say, okay, we have 10 structures, we will take that one. But this is a sad story. After all of this, I had just to talk to Bomben, who is the developer of all this, to ask whether actually this parameter was a good parameter to be used, and it was like, no, we will have to remove that parameter from the spreadsheet.
It's written on the web page.
Yeah, but it was they wrote it later.
That's fine.
So
okay, thank you.
Yeah.
Thank you.
Thank you.
Thank you. It seems we have no more questions, so thank you again. And we move on to our final speaker of the day, Rodrigo.
Hello. Hi. Quero. Oh. Yeah, one moment. Yep. How can I pass? Yep, I'm ready. Hello, everyone. Thanks to be here. Thanks, Fernando and Ralph, to the organizer. It's a very pleasure to be here talking in ICTP. I will present this work that I've done during the COVID-19 pandemic. This work called the Walking Cell, a computer simulation study for metastasis cells. I hope you enjoy the presentation, and I will show you some different simulations, some molecular dynamic simulations that I have done during the last days. Well, the idea, what I show today is the idea that we published together with the guys that are from Mount Sinai, Mount Sinai in the United States. And the idea of this work was to study cells. The guys are experimentalist guys that have done some experiments and send the experiments to publish in the Nature. And the referees told, oh, your job is very well, but we need some theoretical or some simulations to explain what you are seeing in the cells. And the first question was what kind of cells? What cell I'm interested to simulate? I will look in here. I think it's better. The idea was to study stem cells. The stem cells initially has only one nucleus, and anyone here knows that the stem cells goes to another kind of cells in our body. Yeah. What we will show today is for only mononuclear cells, and all the simulations that can be done with multinuclear cells are open, and I hope that anyone here wants to study this kind of simulation. Okay, the idea was how can I create a model for cell simulation? Yeah. The idea was, I'm a guy that study molecular dynamic simulations from our old school, and I started to create simulations using Monte Carlo, Wang Landau, and many simulations like that. What I can show you here is the idea behind all molecular dynamic simulations. Yeah, you start from an unknown, unstable configuration. You giving some equilibration, and if you have a equilibration configuration, you start to do measurements and things like that. You need to select what the scale you are working. You can perform simulation with atomic simulation, all atoms, a mixed of atomic simulation with coarse graining, or go to coarse graining ones. And after you have a stabilized simulation, and you go to do measurements. You go measure factoric structure, all things, duration, radials, and another kind of measure. And we are now in the machine learning era, so mainly people want to measure the things that machine learners do. Yeah, I don't have done many measures of machine learning, this kind of simulations yet. Okay, the idea, the first work was we need to create a model to simulate cells. What I did, I go to internet and look to images of cells, like this one. You can see a membrane, a nucleus, and actin filaments. So I reproduce this structure in my coarse graining model. Yeah, and we have a membrane, the nucleus, actin filaments. Some actin filaments is connected, and another is living. It's not connected to the nucleus. And the head of the actin filament, it's a very important guy. You painted that it's a gray. The head of actin filaments, we control how this head connects to membrane. How can I control this? I will tell you in the next slide, but this is the main idea. The guys was creating some stem cells, delivered some drugs, the plexin, and measure using IFM probe. They are experimentalist guys, and measure the force to push or pull some cells from that agglomerate of cells that they created experimentalists. So I did the same. I create the same IFM probe, and start to study and push and pull cells using this probe. Yeah. The main idea is, I need to understand how the cells work. I create my model, take this in one unstable configuration that I have created, and using a molecular dynamic simulation with a dissipation term that remove energy, look to how and when all the energy or kinetic energy were removed of the end configuration. What's the final configuration of that cells that I model with some force field that we'll show in some moments. The idea here is experimental simulation, or experimental, I can think it's a simulation, with white type cells, cells with small plexing. I think it's moving. Can play again. A cell with small plexing and cells with a lot of plexins. Here's my model. We can see that when I change parameters of my force field, I don't know how the effective number for this parameter, so I create simulations that are relative. I create the membrane, it's my 1, and changing the parameters for the force fields up this one and down this one. Yeah. And we can see that we can many kind of cells and configurations. Yeah. You see kids, that another one, you see that the cell from this kind of parameters, the cell it's not connected. This is very connected one. And here are the atomic force tip. And after some time, I start to push the cell using this atomic force. I can control the base of the atomic force. I'm showing only the tip. And when the tip's touching the cell, you can push and measure the force that I'm doing to pull or push the cell. In this simulation, I'm pull, Yep. This movie is in this reference in the YouTube. Here, I'm trying to understand tissues. So we create a big one. I think that's the internet. It's not. The internet, it's not. Okay. What we will see in this simulation, what these two movies is this cell is a cell with low plexin. So we will see that the movie show that the tissue is like a tissue of bone, of fat bone, a fat tissue. And if you have a lot of plexin, you will see that the structure will form. You stay in a structure like a bone compact tissue. The movie it's not helping. Yep. I will try to return. Yep. This is the process of equilibration. I only don't have temperature. You have removing energy and see what's the stable configuration. Yeah, you see this stay like a very compact tissue, and the tissue is more slowly connected. Okay. What we did after we have this configuration in equilibration situation, I pick one cell in the middle and push. And another configuration, I pick one cell in the border and another cell in the middle. You have to see the difference. It's different to take one cell from the border of the tissue or one cell from the middle. In the kind of simulation, it's not so connected. It's quite the same. The force, it's low to remove. It's quite obvious. But for the case of the simulation, with the tissue, it's very hard. Pushing the middle is very difficult to push in the border. And this result, it's very in agreement with the experiments that the guys performed in the experimental one. The second problem was the walking cell. The walking cell was a very, very exciting problem because I was in my home and trying to take these cells in an environment. And the first thing that I need to put temperature. Yeah, because if I put a cell in an environment, I need to bring them at temperature. I push two walls. One wall is flat and not rough. And the another wall is flat and rough. I put the cell and the cells doesn't walk. When I push the friction or the rugosity in the bottom wall and leave the cell without only entropic force like temperature that the head bath do this rundle shakes and the cells rock. And I stay in my home and I start to screening my wife. What a crazy guy! The cells walk, and I don't understand why, but I'd be very happy. Well, if I want to study, we include the cells in using temperature, we need to understand what's the configuration of the cell from some selected range of temperature, because I don't have, I don't know the parameters that define the temperature of my model, but I need to know if the cells have a simple behavior only changing the temperature. We'll see that for many different parameters that I connect to many different cells, for different temperatures, we'll see the behavior is very different, but the cells stay stable. Yeah. One of my students, it's a graduate student, performed this simulation, changing the temperature, and gives a long time of simulation to see that the simulation are stable, and measure a time of the average of this energy, the total energy of the system. I will take the average, take the average energy for that temperature. We see that it's a linear behavior. the students perform a numerical derivative of this straight line, and we see that the specific heat is quite flat. And I think, yeah, it's nothing amazing, but my cell is still intact. It don't behave, don't have some phase transition or some structural modifications. Oh, okay, I can change the temperature, and you can study it in this regime. Here is experimental experiments performed in the United States. The guys created some microchannels and put cells in one, like an ensemble of cells, and do this movie with some markers and see how the time of the cell migrates from one position to another. And the idea is, well, I need to create a simulation, and I need to create a constraint and see how the cell works. If the cell run, what's the parameters of the cell, how the kind of the cell that walks, okay? You can see that this simulation, the center of the mass of this cell stay in this randomly shaking the position that I put. The average, the modulus of the velocity, the center mass velocities are only oscillating near the zero. But this one, it's a representative cell that walks. You can see that the center of mass go very, 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 very far. And the velocity increase. And I performed some other simulations that this velocity is stabilizing one velocity that the cell wants. Selected and changing from, if I change the temperature, and only show only one representative simulation. We'll see. I put my camera walking with the cell, and we see that the floor are going to another direction, okay, to represent that the cells are really walking. We see that the cells shaking, touching the wall, and with the friction, I push the wall for that one direction and I go to another. Okay. I need to put here. What I did after that, I performed 10 simulations for all group of parameters and measure how the distance that the cell goes far away from the zero position. Okay. And perform average, each point it's average in 10 simulations, yeah. And we see that if we change how they stiffen off this membrane, here is I change the stiff of the membrane. For each curve, I'm changing the connection of the head with the membrane. So why we selected these two parameters for the cell? Because in United States, the headers of the group are a couple. The guy thinks that the membrane, it's that govern the dynamics. The another thinks, no, it's the head that govern the dynamics. So I think I will simulate the both and see. I think that both parameters change the behavior of how the cells walk. The idea is to control how long, if a cell walks a lot, you can connect it with metastasis, because if you can control how long the cell, a cancerous cell, goes run in our body, you can maintain the cancer in that location, don't goes to evolve to metastasis. This is the main idea. Here, it's the boring stuff. I create a classical molecular dynamic simulation. We use the main potential force fields related with polymers. The only thing that have different here is a potential related to the friction. It's not quite normal to think in a potential that generate friction, but during my PhD, the focus of my PhD was to study friction in nanoscale, and it's possible to model a potential that generates some friction. This is the main idea of the potential. It's this one generates the flat potential, and this one is the rugosity. So you push quickly. I think it's my time is over. Okay, so you pass from here. From this is how can I create the code, and only for purpose of showing a spoiler, we are putting this cell now in a vein, thinking that we have a fluid, and we put the cells inside that, and we are interested to see how the behavior of how the cells are walking in a vein. Oh, control. I'm finishing. Oh, we stay here. We are from Juiz de Fora, that stay near in the middle of this triangle, Belo Horizonte, Rio de Janeiro, São Paulo. Here, our team, Vinicius and Guilherme, stay. You see that guys here. And we have a workshop of post-graduation in physics, and if any students want to stay there and meet Juiz de Fora, it will be a pleasure to receive anyone who wants to go there. Thank you. Well.
Thank you very much. Okay. Questions? Yes.
Did you simulate cell adhesion, the connection between the cells, tight junctions or other types?
Here we have we need to, I don't need to put adhesion ad hoc, because when I create a cell, we have the force field. I think that, oh, this cell, it's the same of that. So now this cell has more neighborhoods. So the simulation will connect them, because one membrane atom will see the another membrane atom. Why this is different? I don't see why. And it's the same interaction, yeah?
Also another question, does this behavior also appears if you have polarized cells? For example, one where the nucleus is on one side of the cell and the actins are longer on one side, smaller on the other?
Yeah. This is another spoiler that I can't put here in the presentation, because the guys stay in that moment study how the neurons is formed, and we start to put inside the cell actin filaments, that these actin filaments grow and generates the shape. You are looking in that moment to the shape that one stem cells goes when we are differentiating to be a neuron. And this is the idea of study this actin filament grow. And the next step is the idea that Fernando showed us, put the charge. I know that charge is dictate the behavior of cells, and at that moment we are planning to study with the effects of charge in some position, or how the charge will go to flow to position, change position, yeah.
Are there more questions? I have a question. Your system, when the cell is crawling, it looks very symmetric. How does the cell decide in which direction to go?
Yeah. Here. When I perform this simulation, because this I do 10 simulations of each selected cell. And in the average, the cells go in the 50% from one direction, 50% of another direction. There is no bias. Okay. There is a professor that studied with us and trying to understand why the cells select one direction. Yeah, because we push random velocities in the initial configuration,
Yeah.
why the cells choose to go to one direction. What we telling, we not see the simulation, the results, but we're telling that if there is a small amount of momentum in the initial configuration, we select random generation velocities. So if you don't remove the velocity of center of the mass, the cells will prefer that direction.
Okay.
Right.
Yeah.
It's something like, let's see, it's something like that.
More questions? Well,
Yeah.
you all look very tired.
Yeah.
It was a long day. Thank you very much. Thank you to
Thank
all
you.
the speakers. And